Welcome to Sigest, your fortnightly serving of digestible science from plant and food research.
Kia ora and welcome to another edition of Sigest. Very excited about today's guest, Dr Esme Robinson, who is a real-life ichthyologist. An icky what, I hear you ask? Well, she's a fish scientist. Esme's work is aimed at understanding fish behaviour and personality. Now this might sound like a pretty weird thing to devote your life to, but it has really important implications for sustainability and management of fisheries in New Zealand and also around the world. Okay, welcome to the show, Esme. Thanks for joining us. Most people will be thinking, seriously, how does a fish have a personality? And how do you even study that? Do you take them out to a dinner party and tell them a few jokes or something? I mean, how does this research actually work?
Thanks, Andrew. Yes, I do. I guess if you first look at a fish swimming around in a tank, then they're fairly limited in their ability to display personality. But actually what we're really looking at is the way that they respond to different scenarios that they're presented with, and what are the sort of cues that are important to stimulate changes in their behaviour, and the differences in behaviour between either fish of the same sort, of the same species, but also fish of different species as well. Mm-hmm. So we're sort of looking at the way that they respond to different environmental cues.
What sort of cues do you mean, though? I mean, are we talking things like light or temperature, or what sort of thing? Yes. What sort of cues are you working with?
So with the main species that we've been working on, which are snapper, we've found that actually the visual cues are the most important to those fish, and so we've been particularly interested in looking at the effects of light and how light affects the behaviours that they display. And so snapper in particular are really their behaviour is focused on differences in colour, particularly different coloured lights, and the brightness of the lights that they're presented with, and also contrast between different lights as well. So often we find that fish can be particularly interested in a really bright blue colour, but they might be less interested in a dim blue light or dim blue object. But then some of the fish, if they're presented with an object or a light that's red, can be totally disinterested. So they have differences in the way that they respond to those cues.
Okay, and is that within one species of fish, or would we be comparing like snapper to blue cod or something like that?
Yes, yeah. So one of the sort of interesting aspects that we've found, particularly in snapper, is that it changes with the age of the fish. So one of our early findings of the work is that the younger fish that would typically inhabit sort of estuarine or inshore coastal waters, they're usually living in an environment where the light is predominantly sort of red-yellow light, and those fish, their vision is particularly sensitive to the sort of red-yellow part of the visual spectrum. Whereas as they get older and in their normal environment, they might move further out into deeper water, where the light is sort of blue-green, and then their vision becomes more sensitive to blue-green parts of the spectrum. So in blue-green light, their vision is really sharp, whereas if they're subjected to sort of red-yellow light, then their vision is less sharp. So often the responses change with age within a species, and also we find that if the species naturally inhabit different environments, then also their vision can be different based on the environments that they inhabit.
Okay, so as they change and as they age, they're sort of, depending on the environment they're in, their vision changes as well.
Yes, that's right, yeah.
So, I mean, this sounds kind of a tricky thing to study. How do you actually work with the fish and stuff? I mean, you don't go out there and follow them around, I guess, in the estuaries or at sea?
No. So most of the work that we've done so far on vision is lab-based work, and one of the pieces of equipment that we've used most extensively is called the optomotion machine.
Sounds fancy, yeah.
Yes, yeah. And so what that is just it's based on the optomotor response, and some fish show this optomotor response. It's strong in some species and less obvious in others, but fortunately for us, we've found that snapper have a very strong optomotor response, and what they do is if they're presented with a moving pattern of black and white stripes, then they'll turn, if they can see those stripes, they turn and swim in the same direction as the stripes are moving. So we tend to put them in a round aquarium tank surrounded by a moving cylinder of black and white stripes, and then subject them to, or not subject them, and then put them in the tank. And if they can see the black and white stripes rotating, they'll turn and orient with the stripes and start swimming in the same direction that the stripes are moving.
Wow.
And so what we can do then is manipulate a lot of different environmental conditions and find out how those environmental conditions affect the vision of the fish. So for the colour work, we've changed the colour of the light that's available in the room for the fish. We've done work on changes to vision with turbidity and so on. So we get a lot of really useful information, particularly for snapper that have a very pronounced optomotor response.
So I guess people at home that are listening to this might be thinking, like, why on earth are you going to all this trouble to find out, fish follow these sort of visual responses and things? So what kind of practical applications do you have for this research in the future?
Yes, yeah. So what the work is linked in with, the project that we're a part of, is developing the precision seafood harvesting technologies, which is basically the way forward for trawl fishing technologies. So I guess trawl fishing for the future. And what's really important about the precision seafood harvesting is that rather than a traditional trawl net where a fish would be dragged along behind a boat, in the PSH, the precision seafood harvesting, a fish can swim freely within the trawl fishing system. So the trawl environment keeps the conditions within the control of the animal, and so the fish is able to choose its own pathways within the net. And so what we need to do is we need to understand the cues and the way that the fish might behave within the precision seafood harvesting, and that will help us to develop better selection mechanisms within the trawl system, and also to really show, as it says, some precision in the species that we target within the trawl system, so that if the trawl system is set up to really home the fish in a way in which the fish are less stressed and they're able to swim freely, then they're less likely to experience damage and they're less stressed, and as a result we end up with fish of better fillet quality also.
Okay, so it's about making the fish less stressed, but it's also about trying to sort the fish based on their size, in terms of younger fish will be attracted to a particular light or a particular colour, and older fish will go into a different part of the net because they might be attracted to a different light or colour as well. Is that kind of how it works?
Yeah, so potentially, if we know what the right cues are, what are the things that we need to focus on in the system, then potentially that's the sort of system that we can start to develop. Yeah.
So this is a bit of a facetious question, but there's a lot of New Zealanders out there that go fishing every weekend, I guess myself included, and try and target fish like snapper. Is there anything from this research that the recreational fishing people might be really interested in?
Yes, well, I think based on what we've found in the lab, I think if you're out fishing, definitely, particularly for snapper, they love light, and so I'd really, think about including some sort of, glow sticks or some sort of light attractants for the fish in the fishing equipment. Yeah, they're particularly keen on coming and inspecting light.
Yeah, so they're definitely curious. Yeah. A little bit like us, I guess. We're always attracted to light and things
curious.
like that.
Yeah.
Curious. Okay, now I know this is not the only part of your research. You do a lot of stuff as well. You work also on flounder as well, which is a very different fish, I guess. What exactly are you studying with the flounder?
Yes, so we have a different project running with the flounder, and that project is based down at Lake Ellesmere here in Canterbury. And one of the differences about Lake Ellesmere as a water body is that it's manually opened to the sea at different times of the year, and it's also a very shallow lake, and so it's particularly sensitive to changes in temperature in the lake itself. And so what we've been looking at in flounder is the way that they respond to those changes in temperature within the lake, and whether there's, basically what is the upper thermal limit for flounder in the lake, and also the lower level. So what are the low temperatures that the flounder find it hard to tolerate, and it's particularly important obviously in the face of sort of global climate change, but also because when the decisions are made on whether or not to open the lake, then changes in temperature might be an important part of the decision-making process as to when is a good and when is a bad time to have the lake opened.
Okay, so it helps you to manage that lake and manage that resource by knowing what the fish can handle and what they're happy with in terms of that.
Yes, yeah.
Okay, so that's another thing that sounds like it's quite hard to measure and research. So how do you actually measure temperature effects on a flounder?
Yes, so basically the major part of the work is to find out what's called the aerobic scope of the flounder. And so what we're looking at there is the energy that's available to flounder between the high temperature limit and the low temperature limit. And what we tend to look at is changes in oxygen consumption to work out this aerobic scope. And so what we have is we look at oxygen consumption by rested fish, a fish that's not moving and is rested in rested conditions, and then we look at the maximum oxygen consumption of the fish. So what does the fish, how much oxygen does the fish use at its upper limit when it's been exercised, the maximum amount of oxygen that the fish can use. And then the difference between the rested amount of oxygen consumption and the maximum amount of oxygen consumption is called the aerobic scope, so the energy that's available to the fish for other activities like movement and reproduction and so on. And so what we usually do to get the maximum oxygen consumption in a normal fish, like something like a snapper, is we've got a swimming tunnel here at the university, and we put a fish in there. It's a bit like the equivalent to a human treadmill, and the fish swims along into a current until it reaches sort of its maximum exercise ability. But the trouble with flounder, when if we were going to try and do a similar thing with flounder, is that they're a bit crafty and they just suck onto the bottom of the swimming tunnel. And
sounds like me at the gym. Yeah.
Yeah, that's what I'd do on a treadmill. Yeah. And so whatever, you can inflict them with whatever water current you like, and they'll just suck themselves onto the bottom, and they don't swim into the current. So we've had to come up with a different technique for them, and essentially what we actually do is just tickle them on their tummies, tickle their tummies.
It's very low tech.
Yeah, very low tech. So what we're trying to do is to prevent them from settling on the bottom of a tank. And so if you keep a hand underneath the flounder and keep lifting them from the bottom, then they tend to try and swim away from the thing that's preventing them from settling, and that way we can keep them swimming, usually not for very long, so it usually only takes 4 or 5 minutes for a fish to have exercised for long enough, and with the tickling method.
Yeah, just to keep them active. Yeah. So, are there any results you can talk about so far from the study? Like, how adaptable are flounder to these changes, particularly in the light of, I guess, things getting warmer and warmer?
Yes, actually, we've been quite amazed, really, with the ability of the flounder to adjust to a range of temperatures. So we've worked so far at about 9 degrees, and the flounder have been had a reasonable aerobic scope, so energy available there. And then we're just at the moment looking at fish closer to sort of 20 to 22 degrees, and the fish are still showing reasonable ability at those temperatures too. So that's quite a wide range of temperatures that the fish are able to tolerate and to survive in. And so in a way it's not surprising because of the environment that they come from, that is, that it's something that they experience in their natural environment, but it certainly does make them a particularly tolerant species.
Yeah, so they're quite robust, but that might not extend to other species, I guess, or, could you say that it would be the same for another species, or is it just because they are a bit unusual in the environment they're in?
Yes, so it is always hard to know. I mean, generally the environment that the fish comes from gives a good prediction of what temperatures they might be able to tolerate. So fish that come from a much narrower temperature regime would be expected to have less ability to tolerate temperatures. So in the future we are hoping to extend the work to some of the other species found in Lake Ellesmere too, and I guess sensibly we would expect that those fish also had a similar ability to tolerate temperatures. But I guess one of the things in a natural setting is that fish have ability to be able to migrate away from temperatures that they are not suitable for them. So there's thought that potentially fish in Lake Ellesmere, when it starts getting hot in the lake, may migrate to different areas of the lake that are a bit cooler. So that's a sort of a different side to the way that fish handle changes in temperature also. So some fish, even though they might live in an environment where the temperature varies quite dramatically, the actual temperatures that they experience could be more narrow than predicted just by measuring the lake temperature.
Yeah. So it's pretty important research. We do understand very little about physiology, I guess, in the wild and stuff. So it's really good to get that information out. Just to finish off with, Esme, we'll just talk a little bit about you, the scientist, because it's always interesting to get a bit of a background to the story. I guess fish is a slightly unusual thing to get into. How did you end up studying fish?
Yes, well, I think when I was thinking about what area to do my postgraduate work in, I think the idea of working on fish appealed to me because it has an industry associated with it, and
Sure.
obviously the fishing industry in New Zealand is very important, and so I was always keen that the research that I was undertaking would have some practical sort of aspect to it, and that any research that I undertook would actually have some relevance to New Zealand as a whole.
Yeah, so it's that whole applied angle, I guess.
Yes.
That's very true, I guess, for the area you're in. And so you did your PhD in this kind of research before, yeah?
Yes, so that's how I initially started with the work. So I did my PhD at Canterbury University, and I was very lucky to be able to work on Antarctic fish as a part of my PhD project. And in the PhD, we were also looking at temperature, and actually the Antarctic fish make a good comparison, I guess, with the flounder, because the temperatures that they experience in the Antarctica, although they're extreme, they're very narrow, so there's very little variation in the sea temperature in Antarctica. So these are fish that we worked on in Antarctica had a very narrow ability to tolerate changes in temperature, which of course is going to be important potentially for them if the sea starts to warm as a result of climatic change.
So you actually got to spend some time in Antarctica, is that correct? You actually got to go on the ice?
Yes, yeah. So I had 3 different field seasons in Antarctica, a couple of months each time, which was a wonderful experience. A really very different sort of place to work in, but a very enjoyable experience each of the times, yeah.
It's a completely different environment to how most PhDs. Mine was spent in a lab. I'm incredibly jealous of your time you managed to spend down there. Okay, so when you talk about the temperatures that these fish are living in Antarctica, what sort of temperatures are we talking about? Yeah,
so it's reasonably cold temperatures actually. So for the fish that we were working on near Scott Base, the general temperature for them would be around -2 degrees, and it's not frozen at that temperature because of because it's salt water, so it has a slightly lower freezing point than the fresh water. So generally they're about -1.8 degrees for those Antarctic fish that I was working on. And the particular species that I studied for my PhD was what we considered pretty tolerant of temperature change for an Antarctic species, but actually that translates to them only being able to tolerate temperatures around 4 degrees was their upper limit. So I guess that's a range of sort of around 6 degree change in temperature. So very narrow in comparison to something like a flounder, and only really minor changes in their sea conditions could actually be quite important for the future of those fish.
So they're quite finely tuned then. They're not really happy outside a very narrow temperature band.
Yes.
Sounds like some people in my open plan office.
Yeah.
So as you, I guess, the question I ask scientists is like, what actually motivates you to get up in the morning and do your job? Because I guess it's probably not the money, let's face it. Is there something that's kind of your passion in terms of science? I mean, you've already talked about the applicability of your work to industry. So is that the main driver, or?
Yes, I think it's a combination of things. So I think, yes, particularly that I'm very pleased that there's a reason for the work that I'm doing, and that I know that I'm contributing to some really important changes and developments in fishing, and also in the protection of different fish species, particularly in Lake Ellesmere and so on. But also just the intellectual challenge of understanding something that's new and working out, finding answers to questions that are new. And I think it's always nice to come into work and have to actually think about what I'm doing, and although we're doing similar experiments each day, there's always something new that we're finding out that we didn't know before we started.
Yeah, I guess it is an unusual job, as you're always, I guess, trying to find new things out and do things differently, and on that edge of discovery is an exciting place to be, I guess.
Yes, yeah, definitely. Yeah.
Okay, well, thanks for talking to us today, Esme. It's been fascinating learning about your work with fish behaviour and also with environmental, responses of flounder to temperature and stuff like that. And we've learned a lot about fish biology, and it's been really great having you on the show. So thank you very much for talking to us.
That's great. Thank you very much, Andrew.
Okay, you've been listening to Dr Esme Robinson from Plant and Food Research, who is doing some pretty innovative and creative research around understanding how fish behave and how they respond to changing environments. So I won't flounder around any longer. I'll just scale back the talking and leave you to mull it over. Hopefully you've also been hooked on this awesome research. Thanks for listening. Make sure you tune in again soon to our next SciGest podcast.